Perfect. Thank you, Marta, and thank you for all the others, and also for the master to give me the possibility to tell to the other, in particular to the master students, about this specific topic that is paleoneurology. As Marta told before, is my topic for the PhD, but exactly what is paleoneurology? Came from this discipline is inside the paleoanthropology, so the study of the human evolution, and in particular came from three specific words. Paleo, neuro, and logia, that came from the ancient Greek and means ancients, nerves, and studies. Obviously, so the paleoneurology is in general the study of the brain evolution. In our specific topic, instead, so in archaeology and paleoanthropology, we focus the attention on Homo, so in the genus Homo, and all the other genus and species linked with our evolution. So, for example, with the Australopithecine or the Paranthropus. So, doing a resume of what study the paleoneurology, the main organ and the main topic of this field is the brain. That is the main organ also of all the system. It's made up and is located in the cranium and is part of the central nervous system. It's very defended, protected, as we see. In fact, for example, it is protected by 20 bones in general, in a specific way by the neurocranium, that is the posterior area, posterior bones of our cranium, the famous flat bones, like for example the frontals, parietals, occipitals bones, and the sphacnocranium, that is instead located in the frontal and so anterior area of our cranium, is the so-called face. The brain in this case is connected with all the other system of the nervous system with the brainstem that we can see here. Is a big rib we can see, so there is the organ and this structure that connect the brain with the peripheral system. And is useful also in this case by an evolutionary point of view, this hole that is called foramen magnum, because create the possibility to the brain to have this connection between the central system and the peripheral nervous system. Also is important in Homo sapiens, so right now, the volume of the cranium of the brain is around 140,000. Is important because we can see that the body mass, in this case, of this organ is around the 2%. So it's a small organ. But if we see instead the consumer and the energy that spent this organ, we see that there is a problem because the brain instead use the 15% of the cardiac flux and also the 20% of the metabolic system, the metabolic resources. This means that this small organ have the possibility to spend a lot of our energy. In others, we can see that the brain is stratified. I suppose that you know about the white and the gray matter. The white, in this case, is this, and is the structure of the brain, is made up by glial cells, are very simple cells without nervous and cognitive abilities, but in this case works like, for example, the protection, but also nutrition for the gray matter. The gray matter instead is the external surface of the brain, and is here where the high cognitive abilities take place. About this surface that you can see in this case is colored in green, red, and yellow, is called this pattern duroencephalic. What means? As you can see, there are holes and also hills are called sulci and giri. We can see that this is an evolutionary trick because in a small place, this surface had the possibility to have more neurons and also the possibility to work different and specific cognitive abilities. This is really interesting because it was one of the first characteristics in neurology, paleoneurology, and in general in all the studies that has an object, the brain, was the first characteristic identified as specific cognitive abilities in our species and was firstly mapped. So we can see that not in this image, but in the right image, we can see that all the brain, the area of the brain has a specific name, like for example, the superior frontal sulcus, the middle frontal gyrus, because the first step of these disciplines linked with the brain is trying to create a map of our brain, trying to explain the specific cognitive abilities. Exactly. The brain is so important in our evolution, also for our body, that a protection system that we saw before, that is the bones, but also a second system of protection. In this case, we are talking about the meninges. In this case, you can see this also in vivo. You can see this layer is one of the meninges. It would be the external meninges, the dura mater, are mainly connective tissue. The works of the meninges are specifically three. The first one, the protection from the heart from outside and the movement. The second one is the separation between the brain, so the main organ, and the venous arterial system that we can see here, for example. We need also to remember that the brain works like a car, like a machine. So in this case, the nervous and arterial system works not only for the nutrition, but also for the temperation and for the hot and warm in the brain. At the end, we have the nutrition system. In some specific cases, in fact, the brain can use this liquid to obtain energy. The brain is very complex in its organization, has different kind of subdivision. The main importance is, in this case, in our case, the horizontal brain subdivision. First one, I think that all of us know about the frontal, parietal, occipital, and temporal lobes. There are mainly these. So this is the first one, main division. The second one is the hemispheric system. We know the brain, and we imagine the brain like the left and the right hemisphere, and like in the film, but also in academia, we are used to see the brain like half the artistic brain, and instead the other half like a rational mathematic mind. It's true. What means that in this case, the left hemisphere works more with artistic and oniric data. Instead, the right hemisphere and brain hemisphere works more with rational data and mathematics data. We can see also that before we analyzed a map, we can call it like a map of the brain surface, only by a topographical point of view. In this case, instead, we have another subdivision. It works not by topography, but in this case, by a cognitive function. What means that in this case, the prefrontal association area, so all this area that is inside the frontal lobes, works only in the creation on our feelings, our mood, and also in the relationship with the other ones. The most important area in this case about this horizontal subdivision are the primary motor cortex, that is this, and the supplementary motor area that are strictly linked with the movement and with the development of an answer to the senses that the stimulate we obtain from the outside. In this case, we can remember also the Broca's area that is located here between the frontal lobes and the parietal lobes, and the Wernicke area between the parietal lobes and the temporal lobes that are really strictly linked with the influence of linguistic capacities in our species and possibly also in some of our ancestors. Talking about and continue talking about the cognitive function of our brains, we can see we have this main division. In this case, for example, we can see the frontal pole that take consideration also the primary motor cortex, the somatic motor association area, and the frontal cortex. This is exactly what it means. That the organ in this case is divided in a natural way. We create also fake boundaries. In this case, this is an artificial boundaries, also like this, to better understand and interpret the data that we can observe. And after one or better in some of the characteristics that are really important in our brain, in particular in the evolution of this system, are the brain asymmetries. The brain asymmetries are specifically two. The first one is the right frontal leptoccipitopetalia, this one. And we can see, in fact, that there is a protrusion in one of the two poles. In this case, there is a protrusion of the right pole of the frontal pole and a protrusion instead of the left occipital pole. The second case instead is the Jacobinian torque. It is a torsion of the brain on itself, like in this image. The problem is that right now there are a lot of studies and research that try to understand which kind of relationship there is between this characteristic and the instruments of high cognitive abilities in our species, in our ancestor. The only things that we know until this moment is that, like the gyrencephalic pattern, these are characteristics strictly linked with the mammals, and in particular, the brain asymmetry is a very high percentage inside the monkey in general, in the primates and hominins individuals. This what means that, for example, in a whale or in another kind of animals, it's very hard to try to understand and see this kind of variability, so this kind of asymmetry. All these features and all these characteristics can help us to better understand what is the organ, the brain. The problem is, obviously, the brain is not a bones, it's not a is a soft tissue, what means that these don't fossilize. So in which way an archaeologist, or in general someone that works in prehistory, have the possibility to study, investigate this evolution? The first one is with the use of comparative studies. So in this case, for example, we have an imaging of Pampaniscus. We can study the social behavior of some small group and communities in different places to better understand in which way some social behavior can burn and works. The second one instead is also the postcranial remains. In fact, the evolution of the brain works also on all our skeleton. For example, the position of the foramen magnum that change in our evolution in the Australopithecines, for example, is located posteriorly, instead in Homo is totally vertical. Obviously, this change the morphology not only of the peripheric nervous system, but also of the brain. In this case, if you remember before, we saw that the foramen magnum is strictly linked with the brainstem. So this is what means that effectively the change of our body, in this case also of the postcranium remains, change also the morphology of the brain, and in this case of the brainstem. Another information that we can obtain about the brain and the brain evolution came from also the archaeological remains. In this case, I shows a index. Obviously, I chose this not in this case for the complexity on the creation of this tool, but in this case because in the middle you can see that there is a fossil. There are some researchers that say that in this case is totally something happened for the case, but other researchers say that in this case there is a specific symbolic idea behind. So we have a brain evolution and a brain system that gives the possibility to the community of this artifact to not only recreate these tools with a specific technique, but also to show the raw material in base of this fossil. We have also a paleogenomics point of view research. The problem of the paleogenomics is that sometimes these analyses are really expensive, and also problem after we can see the data, the information only after all the analyses we have done. And a lot of time, the analysis that we're doing with paleogenomics are not so satisfied for our interest. Instead, the paleoneurology gives us the possibility of studying the bones, in particular the bones of the cranium of anatomical model humans, so of people actually living, but also fossil, so to better understand the morphology of the brain. The problem of the discipline are mainly three. The first one we yet saw before is that the brain don't fossilize, like the bones, for example. The second one is that in paleoanthropology, and particularly the paleoanthropological remains, are usually fragmented. So it's very hard and complicated to try to study the whole brain of a specific individual. But we have a lot of time to integrate, interpolate data segmented from several individuals of different sites. And the last problem instead is that, as we saw before, there is between the brain and the cranial vault, there are the meninges that cover the whole brain surface. This is what means that effectively we don't have the morphology specific of the brain, but a main morphology. So the paleoneurology and the paleoneurologist at this moment use a specific protocol and in particular study a specific proxy called endocast. That is this. In fact, the brain is the only one organ that continues to grow during the times and working with the pressure left in the inner surface of the cranial vault, its imprint. So this is exactly an endocast, that is the imprint, we need to repeat because it's complex more or less, is the imprint natural or artificial of our organ. This is what means that anyway is not the perfect copy of our brain, but there is between the brain and what we can study a kind of filters. So we need also to remove several data to obtain the real data that we are looking for. Until now, exist three specific kinds of endocast. The first one is a natural endocast. In this case, I chose baby tongue, one of the famous Australopithecines in Africa. The natural endocast are really very rare. I think that in the world compression collection fossil, there are only 3 natural endocasts. They are formed with natural and specific geological and biological processes that create and give the possibility to obtain this, a natural endocast. Until instead 10 or 15 years ago, we're very in trend doing artificial endocast like in this way, using molding materials, latex, plaster, and this kind. The problem is that in this case, this methodology are very invasive for the records. Right now, instead, with the use of virtual anthropology, so with the use of methodology came from the informatics, in particular more work also from the biomedicine, like for example, TAC, micro-TC scan, RMAI, and all this kind, we are able not only to acquire in a digital way the bones, like in this case, but also to recreate and interpolate the surface of the soft tissue. So, like in this case, we started from the surface of the bones in this case, and after we recreated the surface of the organ of the brain, something that right now there isn't. Talking about instead the evolution, so we can see that in the Australopithecine as a very small, characterized by a very small brain, we can see that is around the 25% bigger than anthropoid. This one means that the brains of the Australopithecine is yet in another way, is just a bit far away from the individuals in the community that lived in Africa in the same period, and also with the Pampaniscum, Troglodytes, and all the other monkeys that are living in the war in the new old countries like this. In particular, we see that there is a lunate sulcus that is exactly this in baby tongue. This can tell us that in this case there is an expansion of the parietal area that is in this case strictly linked with the sequential action. This is what means. In this case, the Australopithecine probably had the possibility to understand the chain operateur, so the like for example, in which way they can work as stone tools to create a specific object. We can see after another big step, so another big characteristic in evolution of our brain system in the early Homo. When we are talking about early Homo, we are talking about all these individuals of the same chronology with similar characteristics, like for example, Homo rudolfensis, habilis, and all the others. In this case, we can see, like in the Australopithecine, a short and wide pelvis to have a possibility to get birth at this baby with a big head. In the same times, we have a foramen magnum that in this case is located vertically at the cranium, not like the Australopithecine more posteriorly. The canal capacity is bigger, but not like Homo of our days. We have also a flattened calvarium, and we can see in this way, this is what means that the parietal, the frontal, and the occipital lobes are flattened and also smaller. In the same times, but we can see that there is a volumetric and global increase of our cranial capacity. This one means that yes, the brain became bigger in this period, but the connection and the neurologic connection didn't change more. Another big step instead arrives with Neanderthal. Neanderthal has this big brain. We can see that is more the double of the Australopithecine and also of the early Homo, but it's not important in this case the size. So how it's bigger, this cranium. It's important in this case the morphology. We can see that in this case the brain is more longer, in the same time is longer, in the same time there is an expansion of the frontal lobes in this case, in this way. This is what means that in this case Neanderthal had more space not only in the frontal lobes but also in the parietal and the occipital to develop and answer to the stimuli that receive from outside. In the same times, we have some information about Neanderthal that is confirmed in Sapiens, that there is here a cingulate circus that creates this short and fast connection between the frontal and the occipital lobes. This is one of the biggest steps in the brain evolution in our species, in our ancestor. So remember this movement, this morphological changing that we didn't find in Sapiens, because in Sapiens that we see that there is a reduction of the cranial capacity, but you have two different kind of processes, the globularization and the parietalization. This is what means that the brain becomes round. This gives us the possibility, or better give to the different lobes of our brain the possibility to better communicate in a faster way each other. And at the same time, the process of parietalization, that is exactly this, makes our parietal lobes bigger and bigger. This gives us the possibility not only to increase some specific cognitive abilities like the language, but also to better understand, interpret some specific work, like for example, the napping of specific raw material. At the end, also in Sapiens, we see exactly. Can you hear me?
Yes.
Okay.
Yes.
In the same time, so we can see that this globalization is the hand of the process that we see before with Neanderthal, with the curvature of the dorsal cranial arc. That was this arc that was we see it before in Neanderthal and after in Sapiens is totally underlined and is totally evident. Instead, talking about in which way we can work and in which way we can apply the paleoneurology in some specific case, I decided to bring you and show you the case study from Ciota Ciara cave. Ciota Ciara cave is a cave located in the North Italy, in specific in Piedmont. The datation is very, very old to be a Neanderthal site. In particular, there is these stone tools of mod 3, so it's complex. And we found during this year several human remains, in particular 70th and an occipital bones. The occipital bones that I will show you now. The external surface, okay, we can see this, that is the classic chignon of Neanderthal. Is a characteristic only of this species. This instead is the lateral surface. We can see these edges are not very well formed. These give us a lot of information about the age of this individual. Probably was a subadult around 15 years old, no more. The other information came from the internal surface. As you can see, the effectively remains didn't give us a lot of information. You can see there is here a structure and also here. So we decided to focus our attention on the inner surface to better understand which kind of information can give us. We firstly obtained this kind of 3D models, can give us a lot of information. We can see more this chignon, that is characteristic of Neanderthal, and all the other features of the cranium of the samples. You can see here the exact location of this bone in a Neanderthal brain. So our study was before digitalize and acquire in a digital way the sample, and after try to understand in the big confusion of the Middle Pleistocene where these places and this sample could be located. We have done this following three main way research: the description of the anatomical structure, the study of the symmetry, and at the end the prediction of the endocranial capacity. You can understand that these information are really hard to find by a fragment of these sites. The comparison collection was made up obviously by a fossil hominin collection coming from the Musée Naturel, the Storia Naturale of Paris, but also from Rovira e Virgili or Atapuerca, coming from in general from Sud-Est Asia, I think also, yes, because there is Nangong. We have also an anatomical modern human collection, because obviously we needed to understand the variability on the brain. So the methodology was more or less quite easy. We divided qualitative analysis, so the anatomical comparison in this way. This is a methodology coming from medicine and biodata analysis. So no in paleoanthropology, archaeology, something already new. And instead, by the other point of view, we analyze the data with a quantitative analysis in a specific way using geometry morphometric analysis to obtain after the prediction of the cranial capacity. By anatomical description, we can see that in this small fragment that is not bigger than 7 centimeters, we identify the internal occipital crest, that is the crest that divided the two hemispheres of our brains. We identify also the left occipital lobe and the right occipital lobe, the cerebral lobe, the right and the left, and all the other substructure. But the cool things was also that we have the possibility with a specific filter to identify the girification pattern. In 7 centimeters and with the arterial system, in this case, this is the arterial system that was between the brain and our negative, we were able to identify anyway the transverse occipital sulcus, this, that we identify also in the other side. And in the left side, we identify also the lateral occipital sulcus that we saw before and the retrocalcarine sulcus. Are really amazing because individuals and samples from this chronology are not really easy find and spot this kind of characteristic. The dural venous system instead, so the study of the drainage of our brain and also of the resource system and the nutrition of our brain give us a lot of information. We understood that the anatomical model humans has a very high variability, but not like Homo neanderthalensis. Sapiens follow more or less a same pattern that is to the right, so we have this structure like in Ciota Ciara to the right. Instead, in Homo neanderthalensis, we have a very high variability. But we understood also that it strictly linked this pattern to the occipitopetalia pattern. that it was something that before we didn't know. After we decided to create a specific landmark configuration, so with the use of geometric morphometric analysis to better understand the morphology of this brain, we understood that Ciota Ciara were located, in this case, in the HR2 PCI, and we can see that is inside the variability of anatomical model humans, but at the same time it's very far away from the green that is Heidelbergensis, on the blue that is Neanderthal, and this was totally strange. Also, in this case, we see that Ciota Ciara is inside the variability of all the other fossil species, but is at the boundaries. This what means that the morphology of Ciota Ciara was similar to the other ones, but just a bit different and also archaic in our case. So we decided to study not the symmetric pattern, but the asymmetric pattern with semi-landmark configuration. We understood effectively that Ciota Ciara is inside the variability, so it's a brain of the coming from the occipital lobes, but in the same time, the morphology, in particular the asymmetry of this piece of brain, is totally opposite to other the fossil record. This can give us information about also the age of this individual, so the chronology, if it's an early Neanderthal, a young Neanderthal, or a classic Neanderthal. After we used all this data to try to understand better what could be the cranial capacity of this specimen, and we saw that in this case, the Ciota Ciara has a cranial capacity lower and smaller than all the other samples, but we need to remember that this is a subadult, so makes sense. In all the cases anyway, the cranial capacity is able and makes sense, taking into consideration that Ciota Ciara is a subadult, as is also a Neanderthal individuals. So at the end of all this work of paleoneurology, we were able not only to identify and describe this specimen like a subadult, a specific Neanderthal individual, and we described also the morphology of the brain. We have more or less an idea in which places we can put this specimen in our European context of the Middle Pleistocene, obviously. We have also the possibility to study the dural venous system drainage, the asymmetry and asymmetric pattern, and then also by a fragment of no more 7 centimeters, we are able to investigate the general cranial capacity. So with paleoneurology, we were able to do a lot of things by very small fragment. Thank you for the attention.